Hi, you're made of data, not the kind you post online. The kind that's been running inside every single cell of your body since before you were born. And here's the wild part. Scientists figured out how to read it. All of it. And then they realized there's so much of it, no human could ever make sense of it alone. So they built computers to help. That's bioinformatics. It's what happens when biology gets so complicated that you have to call in the mathematicians, the computer scientists, and a few sleep-deprived grad students to sort it out. And today, we're going through all of it. Buckle up. Let's start with the fundamental problem. You are roughly 37 trillion cells. Each one contains your entire genome. Your genome is about 3.2 billion base pairs long. If you printed it out in 12-point font, it would fill roughly 1.5 million pages. That's one cell. Now multiply by 37 trillion. Nobody is reading that. But here's the thing. Hidden inside all those letters is the instruction manual for literally everything about you. Your height, your disease risk, why you can't digest milk, why your hair does that thing. And for a long time, we couldn't read any of it. Then came the sequencing revolution. And then came the data problem. Because once you can read DNA, you realize very quickly, reading is the easy part, understanding it, that's the whole field. Bioinformatics is the intersection of biology, computer science, and statistics. It's the toolkit scientists use to store, analyze, and extract meaning from biological data. Mostly that means sequence data, DNA, RNA, proteins, but also structures, networks, evolutionary trees, drug interactions, gene expression, cancer mutations, all of it. If biology generates the data, bioinformatics figures out what it's saying. Think of it this way. Biology is the crime scene. Bioinformatics is the forensics lab. You can have all the evidence in the world, but without the tools to analyze it, you've got nothing. Everything in bioinformatics starts with sequences. A sequence is just a string of characters. For DNA, those characters are A, T, C, and G, the 4 nucleotide bases, adenine, thymine, cytosine, guanine, right? Sure, except your genome has 3.2 billion of them, and they're not random. Inside that string are genes, sections that code for proteins, regulatory regions that turn genes on or off, repetitive elements, junk DNA, stuff we still don't fully understand. The first job of bioinformatics is to work with these sequences computationally, store them, compare them, search them, align them. And that starts with sequence alignment. Imagine you have 2 DNA sequences, one from a human, one from a chimpanzee. They're not identical, but their close DAC-SEC alignment is the process of lining them up to find the similarities and differences. Why? Because similarity implies shared ancestry, and differences tell you something changed. Maybe that change is harmless. Maybe it's what separates you from a chimp. Maybe it's a mutation that causes cancer. The most famous tool for this is BLAST, Basic Local Alignment Search Tool. You take a sequence, throw it at BLAST, and it searches a massive database of known sequences to find the closest matches. It does this billions of times a day for researchers around the world. BLAST doesn't find perfect matches. It finds the best possible match given the messiness of real biological data, because evolution doesn't care about clean data. It just mutates things and sees what survives. Okay, but how do you actually align 2 sequences? Let's say you have ATCGT and ATGGT. Similar but not identical. You can line them up and see there's one mismatch at position 3, easy. But real sequences are thousands of bases long, and they've had millions of years to accumulate insertions, deletions, and substitutions. So you need an algorithm. The classic approach is dynamic programming, specifically the Needleman-Wunsch algorithm for global alignment, aligning the full length of both sequences, or Smith-Waterman for local alignment, finding the best matching region within longer sequences. Both work by building a scoring matrix. You assign points for matches. You subtract points for mismatches and gaps. Then you find the path through the matrix with the best total score. That path is your alignment. It sounds simple. And conceptually, it is. But do it for sequences millions of bases long, across thousands of organisms, and suddenly you need supercomputers just to keep up. Here's something that doesn't get enough credit. Before you can analyze biological sequences, you need somewhere to store them. That's what biological databases are for. GenBank is the big one for DNA sequences. Maintained by the NCBI, the National Center for Biotechnology Information, it has over 200 million sequences. And it doubles in size roughly every 18 months. There's also UniProt for protein sequences, PDB, the Protein Data Bank, for 3D protein structures, Ensemble for annotated genomes, RefSeq for reference sequences, and dozens of specialized databases for everything from viruses to metabolic pathways. These databases are not just storage. They're annotated. That means alongside every sequence, there's information. What organism it came from, what function the gene has, what diseases it's associated with, what other sequences it's similar to. Annotation is its own enormous field of work in bioinformatics, because a sequence with no context is just letters. Caxect is everything. Let's talk about how we actually get these sequences in the first place. For most of human history, we couldn't read DNA at all. Then in the 1970s, Fred Sanger developed a method to sequence DNA chemically. It worked, but it was slow, painfully, brutally, expensively slow. The Human Genome Project, the effort to sequence the first complete human genome, started in 1990. It took 13 years, it cost roughly $3 billion, and it involved thousands of scientists across 20 institutions in 6 countries. For one genome now, you can sequence a human genome in about a day, for around $1,000. That's one of the fastest cost reductions in the history of technology. Faster than Moore's Law. This happened because of next-generation sequencing, or NGS. NGS doesn't read DNA one letter at a time. It reads millions of short fragments simultaneously, in parallel, and then uses computational methods to assemble them back into a full genome, which brings us to one of the core challenges in bioinformatics. Here's the problem with NGS. The machine doesn't read your genome from start to finish. It shreds it into tiny fragments, typically 150 to 300 base pairs long, reads those fragments, and spits out hundreds of millions of short reads. Your job is to assemble them back into the original genome. It's like shredding 1.5 million pages of a book into confetti, then trying to reconstruct the entire book from the confetti without knowing what the book said. This is genome assembly. And it's extraordinarily hard. The algorithms used are called assemblers. They work by finding overlaps between short reads and stitching them together into longer contiguous sequences called contigs. Then those contigs get scaffolded into larger structures using additional data. The problem is repetitive regions. Large chunks of the genome repeat themselves. And when your reads are only 150 bases long, you can't tell which copy of a repeated region a read came from. It's like trying to assemble a puzzle where 100 pieces all look identical. This is why long-read sequencing, from companies like PacBio and Oxford Nanopore, matters so much. They can read sequences tens of thousands of bases long, less confetti. More coherent chunks, still a puzzle, just a solvable one. Okay, you've assembled a genome. Now what? Now you need to figure out where the genes are. This is called gene prediction or gene finding, and it's not as obvious as it sounds. Only about 1.5% of the human genome actually codes for proteins. The rest is regulatory sequences, introns, repetitive elements, and regions we're still figuring out. So how do you find the genes in all that noise? You look for signals, start codons, ATG. Mark where a protein coding sequence begins. Stop codons, mark where it ends. Splice sites indicate where introns get removed from RNA. Promoter sequences signal where transcription starts. Gene finders use probabilistic models, specifically hidden Markov models, to scan the genome and identify regions that statistically look like genes. They're trained on known genes from related organisms. They're not perfect, but they're remarkably good, and once you've found the genes, you annotate them, linking each one to its known or predicted function using databases and comparative genomics. Here's one of the most powerful ideas in bioinformatics. Evolution is conservative. If a gene has been preserved across millions of years of evolution, if humans and mice and fruit flies all have a version of it, it's probably important. Comparative genomics is the practice of comparing genomes across species to understand function, evolution, and disease. If you find a human gene that's nearly identical to a well-studied yeast gene, you've got a head start on understanding what it does. This approach is called functional inference by homology, and it works. A huge portion of what we know about human gene function was first discovered in model organisms: mice, fruit flies, zebrafish, roundworms, because biology reuses good solutions. Quick recap for context: DNA gets transcribed into RNA, RNA gets translated into protein. Proteins do basically everything. They're enzymes, they're structural components, they're signaling molecules. They're the molecular machines running every process in your body. So bioinformatics doesn't stop at DNA. It follows the whole pipeline. Not all genes are active all the time. A liver cell and a brain cell have the same DNA, but they behave completely differently because different genes are switched on in each. Transcriptomics is the study of which genes are being expressed, actively transcribed into RNA in a given cell, tissue, or condition. The main tool is RNA-Seq. You take RNA from a sample, convert it back to DNA, sequence it with NGS, and count how many reads map to each gene. More reads, more expression. It's a snapshot of what a cell is doing at a molecular level. and it's incredibly powerful. Want to know which genes are activated in a tumor? RNA-Seq want to know how a drug changes gene expression. RNA-Seq want to compare a healthy tissue to a diseased one at the molecular level. RNA-Seq, the data it generates is enormous, and analyzing it requires sophisticated statistical methods to separate real biological signals from noise. If transcriptomics tells you what genes are being expressed, proteomics tells you what proteins are actually present. Because RNA levels don't always predict protein levels. Proteins get modified after they're made. They get degraded. They interact with other proteins. The actual protein landscape of a cell is its own story. Proteomics uses mass spectrometry to identify and quantify proteins in a sample. You basically break proteins into fragments, measure their mass, and use databases to figure out which protein each fragment came from. The resulting data maps the entire protein population of a cell. And bioinformatics is what makes sense of it. This one deserves its own spotlight. For decades, one of the biggest unsolved problems in biology was the protein folding problem. Proteins are chains of amino acids, but they don't stay as chains. They fold into complex 3D shapes, and the shape determines the function. A misfolded protein can cause disease, Alzheimer's, Parkinson's, cystic fibrosis. Predicting the 3D structure of a protein from its amino acid sequence alone had been an open problem since the 1960s. Then in 2020, DeepMind's AlphaFold cracked it. AlphaFold is a deep learning system trained on known protein structures. It predicts the 3D structure of proteins with accuracy rivaling experimental methods. This was a genuine scientific earthquake. AlphaFold has now predicted structures for over 200 million proteins, essentially the entire known protein universe, and it's all publicly available, for free. If you want to see what a protein looks like, you look it up. It's hard to overstate how much this changed biology. Let's zoom out for a second. One of the oldest questions in biology is, how are organisms related to each other? Phylogenetics answers that by building evolutionary trees. In the old days, you did this based on physical characteristics. Now you do it with sequences. You take the same gene from 100 different species. You align the sequences. You build a tree based on how similar they are. The more similar two sequences are, the more recently they shared a common ancestor. These trees, phylogenetic trees, are now built from whole genomes. They've rewritten our understanding of the tree of life, revealed that fungi are more closely related to animals than to plants, shown that archaea are a completely separate domain of life, helped track the evolution of pathogens in real time, including SARS-CoV-2 during the pandemic. Bioinformatics made all of that possible. What if you wanted to study organisms you can't grow in a lab? Most microbes can't be cultured. You can't plate them on a petri dish and watch them grow. For a long time, that meant we basically couldn't study them. Then metagenomics came along. Metagenomics is the sequencing of all genetic material directly from an environmental sample's soil, ocean water, gut contents. You name it, you sequence everything. Every organism's DNA all mixed together. Then you use computational methods to separate out the individual genomes, a process called binning. And suddenly you can study entire microbial ecosystems you've never even seen before. Metagenomics revealed that the human gut contains roughly 38 trillion microbial cells, about the same number as your own human cells. Your gut microbiome contains 150 times more unique genes than your own genome, and we're only beginning to understand what those genes do. Back to humans. One of the most clinically important applications of bioinformatics is variant calling. Every person's genome is slightly different from the reference human genome. These differences are called variants. Single nucleotide polymorphisms, SNPs, are the most common type. A single letter change in the sequence. Others are insertions, deletions, or large structural changes. Most variants are harmless. Some cause disease. Some increase risk. Some are protective. Variant calling is the computational process of identifying these differences by comparing a sequenced genome to the reference. It involves aligning reads, identifying positions where the sequenced bases differ from the reference, and applying statistical filters to distinguish real variants from sequencing errors. Then the hard part, figuring out which variants matter. This is done through genome-wide association studies, GWAS. You take thousands of people with a disease and thousands without. You compare their genomes. You look for variants that are significantly more common in one group. Those variants are statistically associated with the disease. GWAS has identified thousands of disease-associated variants for conditions from diabetes to schizophrenia to cancer. It hasn't cured everything, but it's given us a map. Cancer is fundamentally a genomic disease. It starts when a cell accumulates mutations in tumor suppressor genes, oncogenes, DNA repair pathways. Cancer genomics sequences the genomes of tumor cells to find those mutations. And bioinformatics determines which mutations are driving the cancer versus which are just along for the ride. Those are called driver mutations versus passenger mutations. Identifying driver mutations guides treatment. If a tumor has a mutation in a specific kinase, there may be a drug that targets exactly that kinase. That's precision oncology, and it's only possible because of bioinformatics. Bioinformatics has fundamentally changed how drugs are discovered. Traditional drug discovery was basically trial and error. You test thousands of compounds and hope one of them does something useful. Now you start a target, a protein involved in a disease. You know its structure. Thanks, AlphaFold. You use computational methods to screen millions of potential drug molecules virtually, checking which ones are likely to bind to the target. This is called virtual screening or structure-based drug design. The candidates that look promising in silico get tested in the lab. It's still not easy. Most fail, but the hit rate is better. And you're starting with better candidates. Bioinformatics also helps predict drug toxicity, off-target effects, and potential drug-drug interactions before you ever give it to a patient. This is where it all gets a bit science fiction. Modern bioinformatics is increasingly driven by machine learning. Deep learning models are being used to predict gene expression from DNA sequence alone, to classify cancer subtypes from genomic data, to predict splice sites, to identify regulatory elements, to design new proteins from scratch. AlphaFold, already mentioned, is the most famous example, but there are hundreds of others. The reason machine learning works so well in biology is that biology is full of patterns. Evolution has been running the same optimization problem for 3.8 billion years. The patterns are in there. You just need enough data and a good enough model to find them. And right now, we have more biological data than ever before in history, and better models than ever before. The intersection of those two facts is where the most exciting bioinformatics is happening right now. One more revolution worth mentioning. Traditional RNA-Seq gives you an average. You take a tissue, say a tumor, homogenize it, extract the RNA, sequence it, you get the average gene expression across all the cells in that tissue. But cells are not identical. A tumor is not one thing. It's a mix of cancer cells, immune cells, stromal cells, blood vessel cells, all doing different things. Single-cell RNA-seq sequencing. scRNA-Seq sequences individual cells. Each cell gets its own expression profile. Suddenly you can see the heterogeneity. You can identify rare cell populations. You can track how cells change state over time. You can build maps of entire organs, cell by cell. This is the Human Cell Atlas project. The goal: map every cell type in the human body, all 37 trillion of them. It's the most ambitious project in the history of biology, and it runs entirely on bioinformatics. What does it all mean? Here's the thing about bioinformatics. It's not a niche discipline. It's not just for specialists. It has quietly become the infrastructure of modern biology. Every major biological discovery of the last 20 years has had bioinformatics embedded in it somewhere. The COVID vaccines were developed using genomic data sequenced and analyzed computationally within weeks of the first cases. Cancer treatments are being designed using algorithms. Rare disease diagnoses that used to take years are being made in days using whole genome sequencing and computational analysis. New organisms are being discovered not by microscope but by sequence. The tree of life is being redrawn. We're mapping the microbiome, predicting protein structures, designing drugs computationally, sequencing single cells, tracking evolution in real time. And we're just getting started, because here's the real truth about bioinformatics. Biology generates more data every year than any other scientific field, and that gap between data generated and data understood is not closing, it's widening. Which means the field that sits in that gap, bioinformatics, is only going to get more important, more central, more necessary. You are made of data. We're finally starting to read it. And what we find in there might just change everything.